Hello, and welcome back to this tutorial for Astrolabe's Cell Subset Navigator. In this video, I'm going to show how to use differential abundance analysis. In Astrolabe, differential abundance analysis is a tool that visually identifies cellular populations that change in frequency across experiment features. The experiment I have here looked at patients infected with chikungunya virus in both the acute and convalescent stages of disease progression. So the features defined were patient, gender, and time point for acute and convalescent. So if I select time point for differential abundance analysis, a few things are going to happen. In the MDS map, the size of the bubbles still represent the relative frequency of subsets, but now the color is shaded to represent the fold change of each subset across the time point, with the more saturated green and red bubbles representing a greater fold change. The other visualization is a volcano plot, which can also help me pinpoint those subsets with the most significant change in frequency. In both charts, one subset that stands out is the memory B cells, and here we can see that they are not as prevalent in the acute stage of infection, but once the adaptive immune system kicks in the convalescent stage, I can see a marked increase in the bar chart, and almost a threefold increase according to the box plot. Thank you for watching this series about Astrolabe's Cell Subset Navigator. Remember, if you have any questions, you can always contact us at support@astrolabediagnostics.com.